prominently in the light-dark cycle, but also in temperature, food availability, risk of predators, and so on. And this external oscillator entrains an internal timing system, which we all have evolved, not only mammals, but also bacteria, plants, and insects. And in humans and in mammals, there is a hierarchy in circadian clock systems, in these timers, these endogenous timers, with the clock in the suprachiasmatic nucleus, which is a tiny brain region in the hypothalamus, being on the top in the hierarchy. It's basically the conductor of an orchestra, but every other tissue, almost essentially every cell in our body contains also circadian oscillators. The kidney, the lung, hair follicles, blood cells, and so on. These two oscillators, the external 24-hour oscillator and the endogenous circadian oscillator, by the way, this word comes from circadian, meaning approximately 1 day, just saying that the period of this clock is not perfectly 24 hours. So these two couple, and the light-dark cycle mostly entrains or synchronizes the endogenous oscillator to 24 hours. So humans on average have a slightly longer circadian period of on average 24.12 hours. And this is daily reset to make us live a 24-hour life. And this is done by light perception through the eyes. And you may know that it's not primarily the rods and cones being important for entrainment, but a fraction of retinal ganglion cells expressing the blue light photopigment melanopsin. The rods and cones have also their roles, but these retinal ganglion cells directly project to the SCN, suprachiasmatic nucleus, and reset the clock. And depending on the interaction between light levels and endogenous period, which is governed by your genetic disposition, your behavior and physiology will adapt at a particular phase relation and phase angle to the environmental light-dark cycle. And this phase of entrainment, as we call it, is also called chronotype. So for example, if you have a clock which is fast running, as some genetically inherited HER2, which is a clock gene, families have, then you will nevertheless have a 24-hour life, but your chronotype is early. And if you have a slow running clock, your chronotype might be late. It has been, in recent years, has been increasingly obvious that also this synchronization of our clock to zeitgebers, and the light-dark cycle is the most prominent zeitgeber. Food plays also a role, being a zeitgeber. This is important for health. So a well-synchronized high-amplitude clock is good for health. However, as you know, we live in a 24-hour society with a lot of challenges for our clock. This includes shift work, light pollution, jet lag. Oh, no, I didn't want to do this. How can I get back to the normal? Do you see the Referentenansicht now?
Yeah, I think we have Referentenansicht. I think the top one you just have to click on, and then I think it switches.
No, but it doesn't do it. Maybe I just do it again. What I wanted to do is, how can I switch that? Anzeigeeinstellung. Okay. Oh, maybe with yes. What I wanted to do is get my laser pointer. So So this is the challenge for our clock. We have shift work, we have jet lag, we have people in the hospital with irregular light dark cycles. We have the so-called social jet lag, which is the discrepancy between our behavior on work days compared to free days. We often go to the fridge in the night. We have light pollution in the night. And this is all compromising this important process called entrainment. And also, when we are diseased, this can also promote circadian disruption because people are not outside anymore, don't get enough light. The physiology is also challenging our circadian rhythms, the pathophysiology. And there's another aspect. This disease also promotes circadian misalignment. And all in all, it is increasingly clear and supported by a lot of studies from genetic models in mice to environment to epidemiological studies in humans that a well-synchronized circadian clock is essential for health. However, circadian disruption promotes disease, and also disease promotes circadian disruption, and I consider that as a vicious cycle and a self-reinforcing vicious cycle. So what is the challenge? I think it is time for circadian medicine, and I think we should be motivated because we know that the circadian clock is important for health, but we need better mechanistic understanding, which is often lacking, and it doesn't really end up in the clinics yet and in the physicians. We need implementation of this knowledge in clinical practice. This is not coordinated, and it's just starting. In Germany, there's nothing basically in such a coordinated way. So our mission should be to improve the scientific understanding and promote sustainable translation into medical practice via excellent research, training of a new generation of scientists. This is what we just do. Physicians to form a circadian medicine community. And we need a common conceptual transdisciplinary and translational approach and state-of-the-art methods, as well as systematic training and collaboration. With transdisciplinary, I mean we need psychologists, we need biochemists, we need physicians, we need policymakers to make that all happen. So how I see circadian medicine is that it consists basically of 3 pillars, which I call detecting the clock and targeting the clock and exploiting the clock. And with detecting, I mean that we need to have methods and tools to really diagnose the status of the circadian system in humans. And we can do that with biomarkers. And this is basically the talk I'm giving today. What are our approaches to check what the status of the clock is? We can also use variables, and actimetry is very powerful and provides lots of very valuable longitudinal data. Once in the hospital, there's also a wealth of routine clinical data which can be exploited for detecting the clock. Once you discover that a clock is disrupted, and I use the term disrupted as an umbrella term because there's a lot of levels where clocks can be disrupted. It could be internally desynchronized. It could be misaligned to the outside light-dark cycle. It could be misaligned between the cells. Even within a cell, there can be circadian disruption. It can be low amplitude. So circadian disruption is a term we use as long as we don't know exactly the mechanisms, what is going wrong. This is why we need detecting the clock. But if you have a disrupted clock, then we want to repair it. And this is also part of circadian medicine. And we can do that with light therapy. We can use melatonin, which is a hormone, the hormone of the dark. And there's a high density of melatonin receptors expressed in the SCN, so we can use it as basically as darkness in the pill to synchronize the clock and to try to realign it. We can use exercise. We can use time-restricted eating because, as I told you, also feeding fasting cycles act as so-called Zeitgebers. And there are drugs underway which also target clock proteins and may have the potential to shift or the phase or increase the amplitude. What do I mean with exploiting the clock? This is using it for diagnosis and therapy. For example, we know that blood pressure is higher in the morning compared to the afternoon. And this knowledge should be a common knowledge to all the physicians who prescribe hypertensives. So your blood pressure could be over the threshold in the morning, but not over the threshold in the evening. Therapy, when is the right time to take your medicine? Some medications have a clearly time of day dependence, and this gives rise to the whole area of chronopharmacology. So these three levels of circadian medicine exist, and I want to just today look at detecting the clock and give you an example of how we do that by using transcriptional biomarkers. So, as I have told you, we have clocks in every cell. And I spare you with a molecular mechanism, but there is a transcriptional translational feedback loop with clock proteins and clock genes at the core. And these molecular machinery creates rhythms on the molecular level. So mRNA and proteins have high and low abundance depending on the time of day. And this left side is just an color-coded heat map to show you that in macrophages, these are mouse macrophages where we 15 years ago analyzed gene expression of the whole, the transcriptome, so gene expressions of the whole genome every 4 hours. So we harvested mice every 4 hours over 2 full days and measured all the genes, mRNA levels, the transcripts. And you see the abundance of thousands of molecules, of thousands of transcripts in the body is rhythmic, in particular in these macrophages. And green is high and red is low. And you see some genes are high at 4, circadian time 4, others are low at circadian time 4, and so on. So you have specific phases. And for each time point, there's a characteristic profile of genes being high or low. So the time 8 looks completely different than the time 16. And knowing this means if we just harvest at time point 8, we can analyze this profile, and without knowing it's 8, we can predict it's 8 because we know the profile. And this is basically the fundamental approach of how we exploit transcriptional biomarkers to predict the phase. So what we did in a study which was published a long time ago, or 7 years ago, we used blood cells because we were motivated by this mouse macrophage study, which I just showed you here, and said, okay, macrophages, monocytes is basically the same cells from the same origin. We just take a single blood sample, purify the monocytes, analyze the best time-telling genes, and then use a computer algorithm to predict internal time. So basically, you just need a single blood sample. If you go to the doctor, you just take a blood sample, and the doctor analyzes a handful of time-telling genes and then you know what your internal time is. And internal time is not the same as the time on your wristwatch or on the wall clock, because internally, you may be a late type. And if I wake you up tomorrow morning at 7 o'clock, some of you will say, this is the middle of the night. Please don't wake me up. Others who are more morning types are already up and running. So internal time is not the same as social time. And the problem with our health is that we disregard our internal time too often and just live on social time. So to really find out the internal time, we need assays like this, because this transcriptional rhythms are driven by the internal clock and in combination with the sun clock, and the social clock doesn't play a role here. So blood is fine because nurses and doctors can take it, but you cannot use such biomarkers for self-detection. You cannot use it for field studies. So we developed a novel assay, which is based on hair root cells. In hair roots, there are also living cells, and as I told you, every living cell contains a circadian clock machinery. And now I would like to walk you through of how we develop such tests. And basically what I describe now is also in principle done for the blood test. So what you do is, first of all, you need to know which genes are rhythmic in hair roots. And how is that related to internal time? So what we did is we did a training data set, or we created a training data set. We did a study, which we called hair time study, and asked 13 healthy volunteers to take hair samples, which means kind of pulling out hair, about 10 hair with large follicles every 3 hours over 1.5 days. So we got 14 samples. And then in the second day, we determined the internal time. And we did that using an assay called DILMO, which means it stands for dim light melatonin onset. And this is currently the gold standard for your internal time, for your internal phase of entrainment, of how you stand with your internal clock in relation to the sun clock. And what is done here is you take saliva samples, for example, every half an hour for about 6 to 8 hours in dim light in the evening. And then you determine the abundance of melatonin, this hormone of the dark, which is secreted by the pineal gland by either ELISA or radioimmunoassay. And the time when you see it increasing, this is the melatonin onset, so the O, melatonin onset. And this is considered in the field as the gold standard of circadian phase. It's relatively complicated because you need to get samples every half an hour. And the problem with melatonin is if you shine light on the person, which is bright enough, then melatonin secretion is suppressed. So you need to be in dim light. You need to do it in the evening, well beyond your habitual bedtime. So the reason why people don't do it is doctors' offices are closed. It's very expensive. It's basically only done in research. And our aim was to replace that assay by a much more convenient assay. But first of all, we needed the volunteers to sample a time series, and the final goal would be that we just need one sample. But for this, we need first to characterize the rhythmicity in the hair root cells. Here you see we had 13 samples. 13 individuals with a median age of 31, and here are the questionnaires and so on. And here is the Dilmo time, and we had 6 females, 7 males, and so on. So this is for the people who are interested in that. So what does the computer algorithm do when you now have all these genes analyzed? So what we did is, of course, we extracted RNA from these samples and did gene expression analysis and asked the computer to extract, those genes which are the best for predicting the internal time. Because every of these samples got now a timestamp in relation to this Dilmo value, which is the internal time. So we had 14 times 13 samples. Every sample had a timestamp. And then algorithms like Zeitzeiger, Lasso, or partial least square extracted those genes which are best predictable for the timestamp. And this color code means, for example, all the Lasso models identified Arntel as a good feature, and so on. You can just show you a few. So this is Arntel, also called BME1, and these are the 14 times 13 data points from 13 people, 14 data points, and you see Arntel is peaking around 22 hours after Dilmo. And Dilmo is usually at 9 p.m. So Arntel is peaking at about 7 p.m. When you look, for example, at NR1D1, this peaks 30 hours after Dilmo, which is basically 3 o'clock in the morning, and so on. So it's a different phase, and these are only a few examples to show there's quite some variability, but the power is in the numbers. And then now you have that. You can do a so-called leave one out cross-validation. This is often done in machine learning. So you have all your samples. You let the machine learning algorithm train on all the samples but one, and then you ask, you please use that model and predict this one sample. And this is done for the 146 samples. And then you see this is what is observed and with respect to Dilmo. So this is the true value based on the gold standard, and this is what the computer predicts. And you see a very nice correlation between true and predicted in this self leave one out cross-validation assay. But when you look at this, you also notice these error bars, and we put error bars on the dots for to show that we have 3 different predicting algorithms. As I said, Lasso, partial least square, and Zeitzeiger. And you see here, this is in sample number 1, you get these times for Dilmo predictions. As I said, a normal Dilmo is between 8:30 and 9 p.m. And these are the predictions. So they had, for this person or for this sample, the mean prediction would be 9:27, and this is the standard deviation. Some of the samples, however, the predictions don't agree at all. So here we have 4 a.m., 7 p.m., 11 a.m., which gives you a really high standard deviation. So the models are not agreeing, and this leads to these high error bars. And when you now plot the standard deviation of the models, which is this, against the deviation between observed and predicted, you see there's a correlation. If the models don't agree, you cannot trust this value because then the deviation is too high. Which is good because by using 3 models, we have a kind of built-in control by alerting us if that standard deviation is too high. We have to be really careful in trusting the predictions. So we were asking, what is the reason? Why is sometimes the standard deviation so high? So one prediction was maybe it depends on the time of sampling, or it depends on the particular person, because remember we had 13 persons. And now let's first look at the sampling time, and you see the deviation from the truth, the median deviation is particularly high in the evening before sleep, and then it declines during the night and is normal after the sleep in basically almost the first half of the day. And the standard deviation of the models, which is also a value which predicts us whether we can trust, is also lowest at the beginning of the wake phase, and then it increases again and at the end. So we were saying, okay, maybe the best values we get when we take the hair samples in the first couple hours after sleep. So that was sampling time, better in the morning. And subject. So this is now the mean of the 14 samples. We had 13 people who gave us a whole time series, and the mean deviation is, if the mean deviation of these time series prediction is high, we also got a high standard deviation of the models. Meaning, if the models cannot agree on a particular value, this is probably because, and this is our hypothesis, because this person may have not a good rhythm. So if you have not a high amplitude rhythmicity, then it's very hard for these machine learning algorithms to pick the right peak phase. So we were hypothesizing that for those where the deviation is high and the standard deviation of the model is high, maybe they have a low amplitude, these people. And this was our next question. Does amplitude influence accuracy? So first we looked, so what is amplitude? Amplitude is how strong the rhythm is. And there is not a gold standard for circadian amplitude yet in the field. So it's not cortisol amplitude or core body temperature amplitude or melatonin amplitude. So we looked at molecular amplitude and asked first, are the molecular amplitudes correlated? So again, the 13 individuals looking at the amplitude of this gene versus this gene. And for the high amplitude genes, we see a nice correlation between molecular amplitudes, meaning it's not noise or so. So when you have a high amplitude in this gene, you more likely also have a high amplitude in this gene. So we looked at all the correlations between amplitudes of many of the genes which we have. These are typical clock genes, and the bluer the point is, the more correlated are the amplitudes. So this, what you saw here, was NR1D1 versus Kiart. This was this point, which was, yeah. Oh, it should be, but no, NR1D1, this point. Yeah. shouldn't it be dark blue because it says 0.8? Oh, it's maybe R. Yeah, maybe basically there may be a slight mistake here, but you see what I mean. So then we thought, okay, why don't we define our own gold standard for circadian amplitude by just looking at those, and I have marked them with red. We define an amplitude score by doing a mean amplitude of all these red marked genes. And this is our amplitude score. And you see, we have in these 13 people we analyzed, you have a huge variation in amplitude score or in amplitude. And interestingly, when you now look at how good do the models predict, or how consistently do the models predict the phase, then you see those which have high amplitudes, those individuals, the standard deviation between the model is really low, and those which have a low amplitude, the models are not agreeing. So a low amplitude means less reliable prediction, which is basically clear. If you consider the lowest amplitude you can imagine, which is a flat line, how should a machine learning model kind of pick a peak? Since the flat line is never 100% flat, but is noisy data points, one model might pick early, one model might pick late. They all think it's rhythmic, but they cannot agree on a particular phase. So if you have a low amplitude, your model prediction is inaccurate. So now these were all leave one out cross-validation tests to see whether our models are good. But you never must rely on a single data set when you develop a new diagnostics, because you train the new diagnostics on one data set, and then you can, of course, you can do kind of tricks and split the training data in training and test, and this is what we did in this first phase. But the final validation comes from an external validation, so from a completely independent study. Now we did what we all anyway wanted to do all the time. We only took a single sample, measured gene expression here, and also measured the true phase. And we did that with 35 people, so measuring DILMO. And here you see average age 37 years and so on, relatively normal DILMO with between 8:30 and 9 o'clock, 21 females, 14 males. And now the question was, how good are these 35 samples predicted? And let's first look at this table. What we determine is the absolute prediction error, and this is the median. So if we get 1 hour, meaning that 50% of these samples are predicted have a lower than 1 hour deviation from DILMO, and 50% have a higher deviation. You can also say, okay, 45% lower than 1 hour, 75% have an error for less than 2 hours. And you see when we and this is also here, you can see that here, deviation 1 hour about 50%, deviation 2 hours 75%. But what you see here that if we combine all the models, we get about a prediction error of 1 hour. Yes. The question was whether, again, when we look at the standard deviations of the models, we see some data points with really high standard deviations. of the models. And we also observed again that this was statistically significantly correlated to the deviation between prediction and true Dilmo. So we asked, can't we do something about it? And maybe these ones have low amplitude. But since we have only a single sample, we cannot know what the amplitude is. So the next goal was to estimate amplitude from a single sample. And again, we went back to our training data set where we knew the amplitude because we had time series data, and developed an amplitude, correlated the amplitude score with mean expression of non-rhythmic gleans. This is one example, GLE2. And we found several others and combined it into a lasso model where we can predict using a combination of these, that lasso amplitude, which is the model, very nicely correlated to our measured amplitude. And when we now look whether this in the 35 of our, where we don't have time series, only a single sample, these predicted amplitudes from the lasso model, when it is high, then the deviation between the true phase measured by Dilmo and our predicted phase is low. There's again showing us that the lower the amplitude, in this time it's a predicted amplitude, the less accurate the phase prediction is. So we could correct it for this amplitude, and I spare you the details. And when we do that, our absolute prediction error is now 0.8 hours, which is almost as good as our blood test and in the range of how good the Dilmo also is, because we shouldn't even call it error because it means basically the difference between this test, the hair test, and the melatonin test. Since the melatonin test has also errors, it's unclear. Okay, so when we now look at many people, so we founded a spin-off company. I have shown you my conflicts of interest in the first slide, which is called Body Clock, and customers agreed in using their data for research. And this is data from 4,351 people who did this test. And we now see, as similar to as in shown in questionnaires, such as the Munich chronotype questionnaires from Till Rönneberg, that we have a normal distribution of chronotypes or phase of entrainments with a mean of 20.5 hours. And when we now look at, is there age dependence, we see similar things as Rönneberg saw. Young people have a later predicted Dilmo, a later chronotype, and as you age, you become earlier and earlier, and then at the end, you see there are not so many people. It's unclear whether this goes back or not, but that was interesting to us to look at this a little bit more carefully. When you look at males versus females, you see here that there is a slight difference of 15 minutes in males being a little bit later than females, which is also consistent with the questionnaire data, yet not as pronounced as for the questionnaires. And coming back to this, we thought maybe these elderly people don't have to go to work anymore, and maybe they can sleep in, and this makes them later chronotypes, because again, you remember chronotype is the phase of entrainment, meaning it's a state and not a trait. It's influenced by genetic disposition, but it also depends on light levels and also on self-selected light levels. If you sleep longer and don't get woken up by an alarm clock, you see less light in early morning hours. So we looked at working and non-working, and found chronotype depends on work. And when we ask, and this is all age and sex corrected data, do you work or or not? Then the working people have an earlier chronotype than the non-working people, and it's even more short term. If you analyze the data when people sample between Wednesdays and Fridays compared to all the tests done on a Sunday, the Wednesdays and Friday tests are earlier, give earlier chronotypes than the Sunday tests. And intriguingly, if you plot the number of work days against our phase prediction, you see that people who are not working have later chronotypes, and it's almost linearly dependent. And the people working so of course we have the most people work 5 hours a week, and there's a clear dependence on the number of working days, meaning that chronotype is plastic and not rigid. And this can be also demonstrated by looking at me, N of 1, traveling from Berlin to Florida, and I took a hair sample every day here before my trip. And you see this yellow is sunlight and blue is darkness, and the dashed lines is when I was in bed. And this gray time is when I traveled. So that was so I had a Dilmo of about 8 o'clock in Berlin time, and then here I was already almost in Florida, and I still had the Berlin time. And even in the first day in Florida is almost no change, and then in the second day in Florida I jumped by 4 hours and gradually adapted, but I couldn't reach full because then I had to go back to Berlin. And then you see it takes much longer going back to Berlin when you go back to Berlin. So I had really a long jet lag during that time. It will be seen, you see that was 2022, and we haven't done more people in this kind of study. But this is, I only show you that to underline that this chronotype or phase of entrainment is a plastic state which depends on light levels and your genes. So let me summarize. The hair root cell-based chronotype assay is relatively accurate. In particular, when you think about how laborious and costly Dilmo assay is, the hair root cell-based assay may allow amplitude estimation, which we still need to validate with an independent study, which we have not done yet. So care is to be taken here. And the hair root cell-based chronotype assay is useful for field studies because you can ask people taking their hairs and putting it into an Eppendorf tube with a stabilizing solution and mail it in. So people who were customers of the company just mailed it, put their hair in, their hair roots in regular mail, and we this can take a week or so until and the RNAs remain stable. So chronotype is normally distributed, which is now the first time we have an objective measurement of chronotype. And this is confirming data from questionnaires, which were subjective. Chronotype is age and sex dependent, again, also confirming data from questionnaires. And maybe I should have added chronotype is maybe more plastic than we thought, or maybe you guys didn't think it is robust, but I was surprised to see that the dependence on the working time. On the sampling times, there were studies from Martha Merrow when she was still in Groningen, also implying that there's a difference between between weekdays and weekend days. But we also saw it here. So my take-home message is chronotype or phase of entrainment is a state and not a trait. I thank the people in my lab, Bert Meyer, who kind of led the study, Celine in her master thesis developed the hair training data set with Luisa Pilz, who is now a circadian medicine professor at Charité in Berlin. We did all the data analysis of these 4,000 data sets, and with Dieter Kunz, we collaborated in these studies, and Body Clock gave us the data. Thank you for your attention, and I'm looking forward to your questions.